So let us now go into the details of asexual reproduction. That means what are the different types of asexual reproduction that is used or acquired by different types of organisms to increase in number. So the types of asexual reproduction, let us note down here. The first one is fission. The second one is budding. Third is fragmentation. Okay. Fourth one is by spore formation. Spore formation or by special structures. Special structures. Okay. And the fifth one is the vegetative propagation. Okay, the vegetative propagation. Right. So the first one is fission. This all these segments you have already studied in class 10, but the details will be less in class 10. So a little more details, but according to the board, this chapter is not so important for the long answers, 5-mark answers. So 1 mark and 2 mark questions are asked from this chapter. So we will be just briefly going through this asexual reproduction. Now the fission is process. One cell is divided into 2 cells. So the amoeba cell we have studied, the amoeba by binary fission, right? So fission, 2 types are there. One is the binary and multiple. Okay, binary and multiple fission. Binary fission, what happens? The amoeba, one particular cell, it divides. Okay, it divides into 2 cells and 2 daughter cells are produced from the parental cell. Clear? So one cell undergoes the cell division, and after the cell division is completed, it is converted into 2 daughter individuals. So we can see here, after the binary fission is over, we can see 2 cells are produced from the parental cell. So there is no existence of the parental cell after the binary fission is over, or after fission is over itself. There is no existence of parent. So parent doesn't exist. The parent cell only is converted into the daughter cells or daughter individuals, we can say. Okay. So multiple fission, it happens in the plasmodium. Clear? And we have studied that example in class 10 also. And in that, what happens during unfavorable conditions, the plasmodium covers itself with a cyst wall, and then inside that it is converted. So this is suppose one particular cell of the plasmodium. So during the unfavorable conditions, converted into, so cyst wall will be, okay, cyst wall is produced, and the nucleus is converted into many tiny nuclei. Okay, and then cytoplasm will be surrounding the nuclei. Clear? Okay. And when there is favorable condition, the cyst wall breaks and this tiny cells will be going out and developing as new organisms. Clear? But in amoeba, it follows the binary fission as well as a similar process like the sporulation, like the multiple fission, which is also followed by the amoeba, which is called as sporulation process. Clear? So what happens in the sporulation process? So the similar to the plasmodium, during unfavorable conditions, the amoeba covers itself with 3-layered cyst wall. That is called as, the process is called encystation. Encystation is the covering of amoeba with a cyst wall. Then inside that, like this manner, the amoeba divides multiple times and makes tiny cells inside the 3-layered cyst, right? So during favorable condition, what happens? The cyst wall will break and the smaller tiny amoeba, many number of amoeba will move out, right? Like amoeba spores, they will be moving out and they develop into new individuals. So they follow binary fission, and in unfavorable condition, they follow something which is like similar to multiple fission. And the multiple fission process in amoeba is called as sporulation process. So after the encystation, the sporulation process follows. Okay, so covering with cyst wall, then many spores are produced. During favorable condition, the cyst wall breaks, spores move out and they develop into new amoeba. Clear? Okay. Now budding. What is budding? That also we have studied in class 10. So budding, two different examples can be explained. Budding in the yeast, okay, and budding in the hydra, right? So these are the tentacles of hydra and the yeast cell. Yeast is a single celled organism that also shows a similar like process which is called budding. Hydra is a multicellular organism that also exhibits the process of budding. So now what happens in the yeast? So this is the nucleus of the yeast. So during the multiplication time, when it gets mature, the nucleus, it starts dividing by mitotic process. But here what happens, it's not exactly similar to binary fission. You can say there also two cells are produced, here also two cells are. But here the two cells are not produced. One larger parental cell contains a tiny cell like a bud on its body. Clear? So this later what happens, the two nuclei completely separate each other and a wall is formed between the parent and the daughter bud. Clear? So it is not the exact size of the parent. That is why it is considered as a process of budding in the yeast. So this example is yeast cell follows this process. So later what happens, many buds can be produced and they may break from the parental one and they can live into separate organisms. Okay? So what happens in the hydra budding? The hydra is capable of regeneration, right? Some regeneration process we have studied in class 10, right? So what is this regeneration process? The process by which some special cells in the organism's body can develop into any part of the body. They can regenerate any part of the body. So the hydra is also having such specialized cells, and those specialized cells are capable of producing a bud which is exactly a miniature of the parental body. So the regenerative cells, they form first a tiny bud on the hydra body. Later what happens? The tiny bud increases in size, right? And even more it increases. And then it starts developing all the parts which is present in the parental body. Later, when it is completely formed all the parts of the body, then it gets detached from the parental body and lives as a separate hydra. So this is the budding process in hydra. Now the third one is the fragmentation process. The fragmentation is found, the best example for fragmentation is the Spirogyra, right? So the filamentous structure, ribbon-like structure, and it has the cells which are arranged in a linear way, right? Okay. Now what happens here? If the mature Spirogyra is broken into pieces, now each piece will develop into a new organism, right? It will again develop into a new Spirogyra. Okay? So later each of the fragments can develop into a new individual.